Hey, can everybody hear me? Thank you. It's a pleasure to be here, pleasure to be a part of this wonderful set of lectures and symposia. And I want to thank the organizers, and I also want to thank Robert and Patty and Chet for setting the table for me, because a lot of the concepts that were covered, a lot of the data that were covered, in terms of thinking about the behavior of concern for others, how we have positive social relationships, and the underlying biology will be a major theme throughout this talk. And since there's always a danger that I won't make it to the very end, I just want to acknowledge the people who do all the work and those fine people who provide us with the funding to do this work. So really the way that, or what I want to do on this slide, is to give you an outline of the talk, but it's also kind of a bow plan for how our lab works. And the way we think about our lab is it's the neuroethology of primate cognition lab. And usually when you think about neuroethology, you think about exotic critters, right? Like bats and electric fish. Well, I think primates and humans in particular are exotic and specialized as well. We're just specialized for different things. And one of the most fundamental things that we seem to be specialized for is social behavior. Right? And so the way that we approach the lab is to try to take a consideration of the problems that the animals, whether they're rhesus macaques or humans, actually have to solve in their natural habitats. What are the problems that they confront in the physical and social domain? And once you understand what those problems are, you need to identify what the underlying neurobiological solution, the way that the solutions are actually implemented in the brain might be. And so what we spend a lot of time doing is trying to develop paradigms that allow us to bring something of the real world into the laboratory, while at the same time maintaining some control and rigor that allows us to do some neurobiology. And most of that neurobiology is single unit recording from single cells in a variety of different parts of the primate brain, but we also can apply other techniques, pharmacological techniques, stimulation, etc. We try to do at least a little bit of comparative work, but most of the comparisons we do are between rhesus macaques and humans. At a bare minimum, we try to present humans with some of the same kinds of problems, or at least focusing on those sets of problems that they might confront in the real world that are similar to the ones we're studying in monkeys. And some of the time, we can actually employ other kinds of neurobiological techniques like functional imaging. Recently, we have begun to get very interested in individual differences in social function and how they might be implemented by kind of the fine-tuning of these circuits that I will describe. And one of the ways that we have begun to explore this is to manipulate these systems with neuromodulatory hormones like oxytocin. And then finally, we've begun to come full circle and to take what we have learned and to try to apply it back in the field and to see whether we can come to a better understanding of, let's say, individual differences in social function and their underlying biology by going back out to the field, making observations of monkeys who are doing their thing, right, engaging with others, etc., while at the same time gathering some biological data. In this case, various kinds of biomarkers, including DNA that we process for genetic analyses. So, as we all have heard over the past couple of days, humans are incredibly social, right, and not just gregarious, but we do some very odd things from an evolutionary standpoint. We help others, even those that we might not be related to, although from the last talk I can see that maybe that's not quite such a problem. But we engage in all this helping behavior. We form friendships, alliances. We're intensely social. That's also true for a lot of our primate cousins, like the subject of most of my studies, which are rhesus macaques, which also live in large, complex social groups, multi-male, multi-female, characterized by not only competition and the formation of dominance hierarchies where status is important, but also friendships and alliances which individuals can use to better their position. So what is the underlying biology, the underlying neurobiology? Well, at the present time, we kind of have a sketch of something like this from a recent review paper by Tim Behrens and Matthew Rushworth. And the idea is something like this that, whoa, I just blacked it out, sorry. I was going to do that. So, and what we have here are in primary colors. I don't know if you can tell which ones are primary, but the hot red here, the hot blue, green, and yellow, are parts of the brain that we tend to think of as basically generic circuits for reward and decision making. So the general idea is that these areas, in various conditions, whether it's the ventromedial prefrontal cortex, the ventral striatum, parts of the orbital frontal cortex, dorsal anterior cingulate cortex, that these areas basically process information about reward and contribute to decisions about anything. So it can be a decision about whether or not to go to the ice cream bar that we had at lunch, or it can be a decision about whether or not to form a friendship with a particular individual. You're feeding in all kinds of data, and it all gets converted into basically a common currency of value along which you can make a decision. In contrast, there seem to be some other areas, which are the more pastel areas here, the sort of anterior cingulate gyrus, and parts of the superior temporal sulcus on the temporal lobe, which may be highly specialized for social function. These areas of the temporal lobe being specialized for the identification and recognition of other individuals, perhaps their intentions, their likely course of behavior. And this part here of the anterior cingulate along the gyrus has been suggested to be really important for functions like empathy, theory of mind, mentalizing, etc. Now, one perhaps limitation of the current state of the field is that, by and large, most of the data comes from functional imaging studies conducted in humans, where you basically have a person lie down in an MRI machine, and they're going to make decisions, see stimuli, perhaps play an economic game with somebody who's outside the scanner, and then you get something like this, which is an activation map, which shows areas in which there has been increased blood flow, right? This is a hemodynamic signal, which is thought to indicate or be related to the spiking activity or some measure of activity of the neurons within that area. Now, the real limitation here, as anyone who does fMRI will confess, is that it's a sluggish, slow response. It takes seconds to develop, whereas we know that neuronal events take place on the order of milliseconds. And within any one of these blobs of activity lie tens of thousands of neurons. And each of these neurons can have different morphology, as we saw in Chet's talk. They can express different receptors. They can get inputs from different areas. They can project to different areas. So this could be obscuring a lot of diversity in terms of the way that the information is actually being processed within that area. Right? And so this is just meant to illustrate the fact that within any one of those blobs, you have many, many different kinds of neurons that presumably are specialized for computing different kinds of things. So what we do is to try to go beyond that functional imaging approach and go directly to the source, that is the fundamental unit of processing in the nervous system, which is the single neuron. And this is possible, and it's possible to make comparisons between humans and macaques, because I'll assert, and I think Chet will back me up, I hope, which is that for the most part, what we have in our heads is a 30-million-year-old information processing device. So it's all of the brain areas, essentially, that have been identified in humans, more or less, are there in macaques, and they seem to be wired up in more or less the same ways. Our brain is a lot bigger, right? These are not to scale. But beyond that, it's not quite clear where the differences might lie. And this device seems to be a really useful one, right? And it was presumably evolved to enable foraging for food, gathering information that would be useful, and in particular for dealing with others. So in the first set of studies that I want to tell you about, we asked a question of whether we could assess how neurons in different parts of the brain process very simple information about others, that is, visual social information that tells you something about who an individual is, how valuable they might be to you, whether you want to approach them or avoid them. Couldn't be simpler, right? And primates are rather unusual amongst mammals because of the degree to which We all rely on visual information to communicate, and visual displays to communicate social information. So we can quite readily see from this image here, this is a female Homo sapiens. She is young, presumably has high fecundity, high reproductive potential, is not negatively disposed to whomever is looking at her, and so you might want to approach her if you were interested. If you knew something about rhesus macaques, you could tell immediately that this is a male, that he's in good reproductive condition. He's probably got high testosterone levels given the sort of redness around his eyes, and he's intentionally interested in something off the screen. And so you might want to actually direct your attention there. So there's a lot of information that's communicated to us, which we apprehend quite readily. Now the question to us in terms of trying to study how this information might be processed within some of these reward circuits, for example, was to try to come up with a way in which we could ask monkeys how much they valued approaching or avoiding, seeing or not seeing different sorts of visual social information. And as we heard earlier, non-human primates can't talk, at least not yet. So we have to come up with ways that allow them to express this valuation in terms of their behavior. And so this is what we did. This is something that a task we developed and called the pay-per-view task. Basically, a monkey who's sitting in a primate chair looking at a screen, he's going to see a fixation point. Two targets come on, they're visually identical. If he chooses target 1, he gets a squirt of juice. If he chooses target 2, he gets a squirt of juice, but he also sees a picture of a familiar monkey. And within any particular block of trials, sometimes he gets paid more to look away from the picture target, and sometimes he gets paid more to actually see the picture. And then what we do is that in different blocks of trials, we show different sets of images. So one set of images might be the face of female A. Another set of images could be the face of male B. Another set could be the hindquarters, the reproductive skin of females in the colony. And we know that this is a very biologically meaningful and potent signal for males. And in the wild, males spend a lot of their time inspecting these displays, and they obviously have value in terms of predicting potential ovulation time. So we might predict that these different classes of social information might have differential value to male macaques. And what we do is basically, I don't have the slide in here, we analyze the data in terms of how often they choose to see the sets of images as a function of the differential payoff for the two targets. So if they don't care a whit about what set of images is being displayed, then basically they will be indifferent to the two options when they have the same, when they pay off the same amount of juice. If that curve slides to the left or to the right, that tells you that, for example, if the monkey is losing juice by choosing to see the images, this suggests that the images themselves are adding reinforcing value to the juice, right, and making it more valuable, indicating that it has some reinforcing value to the monkey. If the curve slides the other direction and the monkey is actually actively looking away from the image target, right, that means that it's aversive. It's subtracting from the value of the fluid. And when we looked at, this is from a first study that we published in 2005, purely behavioral for 2 male macaques, we find that the value of seeing the female perineum, here in red, is positive. It's consistently positive. And there's also positive value generally for seeing the faces of dominant males, which might seem a bit surprising. You might think that these would be highly aversive, but if you know something about primate behavior, you realize that these are also the animals that you have to keep track of and that you want to have information on. Right? On the other hand, we found that other sets of images, in particular the faces of low-ranking males and the faces of females, had negative value. We don't necessarily understand that yet. But for our purposes, this is really useful because this gives us a metric that we can go into the brain and see whether we can find neural correlates of this social value signal. We actually did the exact same experiment with Duke University undergraduates, where we had them choosing between two options. If they chose, say, the option on the left, they would see a blank screen and they would hear the sound of coins coming out of a slot machine. And the duration of that sound was proportional to how much money they actually won on that trial. If they chose the other option, they would see an image of a person of the opposite sex, And they would also hear the sound of coins coming out of the slot machine. And we basically play the same game. Sometimes they get paid more to look at the set of images. Sometimes they get paid less to look at the set of images. If the subjects ignored the images completely, they could actually take home more money than if they paid attention to the images. Okay, and then we basically play. Oh, and I should say that we had independent sets of raters rate 2,000 images of men and 2,000 images of women, which we downloaded from the website hotornot.com. And they were rated for physical attractiveness. And then we basically divided up the population of images based on those ratings. And we basically can play the same dance here where we look at whether the curves slide to the left or to the right and use this point of subjective equivalence, basically the indifference point in money, in monetary rewards, for each of the sets of images as our measure of value. And what we find is, perhaps not unexpectedly, that for our male subjects, sets of images of women who are ranked from neutral to high attractiveness were positively reinforcing. Okay, so they were consistently giving up a quarter to a half cent per view of seeing these images. Our females were not quite as impressed by the male images, which is interesting in and of itself. And this is despite the fact that these guys in the high attractiveness category were really hot. I mean, they were incredibly attractive, often shirtless, muscular, etc., but they were not motivating. Right? They did not have value. But again, the important point here is that we can now take this value signal and see whether we can find a neural correlate in the brain. And in the first set of studies, what we did is we first ran the imaging study in college males. We identified some parts of the brain that carried information about the value of monetary rewards and the value of these social image rewards. And then in parallel, we went back to the monkeys and we recorded from those same brain areas using microelectrodes. And the areas that we ended up focusing on were the ventromedial prefrontal cortex and orbital frontal cortex, the striatum, and also the posterior parietal cortex, which is involved in actually making the decision about where you want to look. And our imaging study, just like every other imaging study that has been done on this basic question, identified a number of hotspots, including the ventral and medial striatum, the ventromedial prefrontal cortex, orbital frontal cortex, and a number of other areas in which the hemodynamic response scaled positively both with the monetary reward that subjects could win and also scaled with the value of the images that the subjects were seeing. So this is consistent with the idea that this is a generic value system, that basically you feed in information about whatever it is that you are considering, whatever commodity it is, whatever you are going to make a decision over, and its value is represented in the system, and basically you compare the value of two options and you make a decision. When we put electrodes into these areas, for example, in the ventromedial and orbital frontal cortex, we found a striking difference, at least in the monkeys, which is that we found completely separate populations of neurons that encoded information about the value of the images, the social images, that the monkeys were choosing to see versus the value of the fluid rewards that they were receiving. So this is in direct contrast with the idea that there is a common currency system, right? Common currency circuit for representing the value of all things, right? So this suggests that, in fact, perhaps a specialized system has evolved that has perhaps duplicated or harnessed somehow this system that evolved to make other kinds of decisions, but to represent social information distinctly and discretely. We found the exact same pattern in the striatum, where we find different populations of neurons represent juice versus the images that the monkey is choosing to see. And finally, when we look at the posterior parietal cortex, where the decision we believe is being made, one of the areas that the decision is being made, we see that the firing rates of neurons associated with a particular part of space are scaled by the value of the image that the monkey expects to see there. Okay? And it is this scaling here that drives neurons to a threshold for actually eliciting the decision or making the decision to look at that particular image. Okay, so that's summarizing a lot of work, hopefully briefly. And I think it's interesting because a very simple kind of social task, whether you want to approach or avoid, see or not see a particular individual, and the value of making that decision, has already revealed some specialization within circuits that we know are involved in reward valuation and decision making. But it's a very rudimentary task, right? So it's not really asking monkeys a very complex question about how they might interact with another individual, in particular how they might interact in a real situation, right? Because monkeys were only working with a computer. And so what we decided to do was to create a new task, which was, for us, we had a bit of trepidation in approaching this problem because we were actually asking monkeys to interact socially while we were recording from neurons in their brains. And so there's an element of reality here, but there's also an element in which we want to keep them safe, not injure each other, right, and not injure our equipment. And so what we ended up doing was having monkeys interact through an interposed system. So we had two monkeys, a donor and a recipient, who were sitting next to each other at 45-degree angles. They each have their own little computer screen. They each have a juice tube at their mouth. We have an infrared video camera that is tracking the eyes of the donor in this experiment, but we now track the eyes of both animals. And for the time being, I'm going to tell you about data in which we have recorded spikes from the donor from 3 different brain areas. And here's the basic idea of this experiment. We wanted to know whether this donor monkey has any concern for what happens to the other monkey. And if so, how's that information represented? Now, from the behavior of rhesus macaques in the wild, you might think, well, they don't do things like share food, they're not so nice to each other. But on the other hand, they do things like form friendships, alliances, they cooperate, they groom each other with no sense that necessarily anything material would come back to them, right? So this suggests that things that happen to other individuals, making something good happen to another individual, might have some value, and we might be able to detect how that valuation is actually represented in the brain. So here's the basic study. We ran both donor and recipient through a period of conditioning, about a week. And all that happened was there's a set of Pavlovian trials, a set of instrumental trials. And in the Pavlovian trials, basically a colored shape comes on, and it's followed briefly by a squirt of juice. Okay? And depending on the shape, the squirt of juice could go to the donor, it could go to the recipient, it could go to both monkeys, or it could go to nobody. Okay? And the instrumental trials are basically the exact same thing, except the donor monkey has to do the equivalent of pushing a button to make it happen. He has to look at the shape with his eyes. After a period of conditioning, we then give the donor monkey choices between options that are matched for what happens to the donor. Okay? And the most interesting one is a choice between a squirt of juice going to the recipient or a squirt of juice going to nobody. In that case, the donor is never rewarded. He never gets a squirt of juice. You would think if it's all about himself, if it's just completely egocentric, he should never even start the trial. And certainly, he should be indifferent to the two options because he never gets a squirt of juice. The other condition is one in which he gets a squirt of juice or both monkeys get a squirt of juice. Again, it's equivalent to the donor if he doesn't care about what's happening to the other monkey. So we can assess whether if the donors are not indifferent in this choice task, this suggests that what is happening to the other monkey has some impact on their own internal computation of the value of different actions. Okay? So in terms of the behavioral data, it turned out to be pretty interesting. This is an index that expresses whether the donor monkey was more likely to choose the option that resulted in a reward to the other monkey or not, or whether he was more likely to be selfish. So we can think of this as pro-social and selfish. When the monkey had a choice between the monkeys, there were several monkeys in this, had a choice between a reward going to the other monkey or going to no one. They overwhelmingly preferred reward going to the other monkey, which is, for us, quite shocking. And in fact, not only did they have a preference for that, but they would perform this action. They would work to make it happen hundreds of times a day. So I don't think you could argue that they just simply did not understand what was happening. They produced that over and over and over again. Curiously, in the context in which they had a choice between a reward to self or a reward to both, they overwhelmingly preferred to be selfish. That might seem like it violates some cardinal economic principles. But to some degree, this makes sense to us because if the monkeys do not necessarily know that they are not drinking from the same source of fluid, right, that it's not a zero-sum game, then this might be viewed as a competitive situation. We don't know the answer to that yet. What we're working on right now is developing quantized rewards, little gelatinized pellets of fluid in which the monkeys can see how much each of them is getting. So hopefully I'll have more to tell you about on that front later. We know this is a socially dependent behavior because monkeys, the donors were more pro-social when the recipient was more familiar. That is, a monkey that they looked at all day, who lived right next to them, versus a monkey that they rarely saw. And they were a little less competitive. And also, dominant monkeys were a bit more pro-social and a bit less competitive than were subordinate monkeys. And this has been seen in a couple of other studies that were similar to this. Okay, so as I told you before, we recorded in 3 brain areas. Again, our old friend, the ventromedial prefrontal orbital frontal cortex, right, an area thought to encode value for decision making. The dorsal anterior cingulate cortex, or the sulcal portion of the anterior cingulate cortex, which we know to be involved in learning and reinforcement. And the anterior cingulate gyrus, which, as I mentioned earlier in this talk, has been implicated in empathy, mentalizing, etc. And we might think that this concern for what happens to the other, this sensitivity to the reward that occurs to the other monkey might have special significance for neurons in this area. I'm going to skip that slide. And so here's basically the data. The orbital frontal cortex, and this is our population of neurons, but all the neurons basically looked exactly the same. In the 2 red colors are conditions in which the donor monkey chose to give reward to himself. And we see that these orbital frontal neurons are strongly activated both around the time of choice and just before reward. And they basically don't have any signal when the monkey chooses to reward the other monkey. Despite the fact that rewarding the other monkey has value for the donor, it's something that he's motivated to do, it's something that he will work to do. This is a purely egocentric signal. It is about directly experienced reward. Anterior cingulate sulcus, on the other hand, was the exact opposite. So these neurons responded whether the donor chose to the option that resulted in a reward to the other monkey, or chose the option that resulted in a reward to nobody, and flatline for rewards to self. This area is concerned with foregone rewards. It has been implicated in detecting errors in a number of other studies, and this suggests that, in fact, it's representing information about what could have been. Right? This is a counterfactual signal. In fact, a few years ago, we published a paper showing that neurons in this part of the brain encode information about what a monkey could have had he acted differently. And the anterior cingulate gyrus, wonderfully, had a signal that was quite different from these 2, in which the neurons responded to the choice to give reward to self and the choice for reward to happen to the other monkey. Okay? So many of the neurons looked exactly like this, with an equivalent response to reward self and reward the other monkey, and basically no response to rewarding no one. Some neurons were selective for rewards to the other monkey. So this is just a compilation across the 3 different areas. This is the proportion of neurons that are essentially egocentric. And we can see that in orbital frontal cortex and the dorsal anterior cingulate cortex, that's a high proportion of neurons. And the anterior cingulate gyrus is more, takes into account what happens to the other animal. Okay, so we can say that they are referenced to an event that's occurring to another individual. And we think that this signal in the anterior cingulate gyrus is actually important in generating the behavior, whether or not to choose an option that results in a reward to another monkey, because if we look at signal fidelity, right here we're looking at the coefficient of variation in firing. Okay, so it's basically looking at how noisy the signals are. When noise is lower, monkeys were more likely to reward the other monkey, and when noise was higher, they were less likely to reward him. And we didn't find that for the other 2 brain areas. So this suggests to us that, in fact, these signals that are being carried by neurons in the anterior cingulate gyrus are actually important for generating the behavior. So it emerges from this that, in fact, the anterior cingulate gyrus is something like a mirror system for reward. And we can think of that as being linked to empathy and these other kinds of behaviors, where neurons respond, in this case, both when a monkey is drinking himself, drinking juice himself, or when he's watching another monkey drink juice. Okay, I'm going to turn to a different question now, which is the question of how do individual differences in concern for the welfare of others arise? Some people are very giving, the nerdy guy here, and some people are not, the cool guy, right? Why is that so? How does that emerge from the biology and the experience that these individuals have had growing up? We could ask a similar question, which is, how do you account for differences between species? Why are some species relatively competitive and others less competitive? Now, as we heard from Pat's talk earlier, we have strong reason to believe that there are neuromodulatory processes that are involved. Oxytocin, vasopressin, a number of other neuromodulatory systems. We know from the work of Larry Young and others that, for example, differences in the topography of receptors for oxytocin, say the localization within the reward circuitry here in prairie voles, seems to be responsible for the strong social bonds, the reward that these animals have for bonding with each other, which you don't see in the non-monogamous, non-pair bonding voles. It's also been shown that allowing individuals with autism to inhale oxytocin, which was believed to get oxytocin actually into the brain, can improve social function in a task like this one, in which basically it's the schoolyard bully task, where you're playing a game in which you can throw balls to different people on the screen, and the objective is to figure out that one of these individuals never throws the ball back to you. They always keep the ball. Healthy subjects are very good at making that detection. Autistic subjects are not. If you let the autistic subjects inhale the oxytocin, they seem to identify that person who doesn't throw the ball back. So their social function improves, and that might be due to, and I'm not going to go into the data here, that might be due to an amplification of the attention that the individual with autism actually pays to the social actors on the screen. Well, we wanted to explore this question further in rhesus macaques, in fact, to see whether we could take an animal who is not a pair bonding animal, who lives in a highly despotic society, and make him a bit nicer. So we wanted to employ the same methods that have been used in humans. And in fact, given the high number of clinical studies that are ongoing now on the use of oxytocin as a therapeutic treatment in autism and other disorders, we thought it'd be important to actually understand some of the underlying biology. Rather than using the typical nasal spray that's used in human studies, we thought this would be difficult for monkeys to tolerate, and of course they have to trust us to engage in these experiments. So what we did instead was to gradually acclimate them to a pediatric nebulizer. And if you've ever had a child that you've had to rush to the emergency room for whooping cough or something like that in the middle of the night, they can learn to accept this quite readily. We trained monkeys that there would be juice in the middle of that for a few days, and so then they learned to put this mask on. Then we nebulize the oxytocin at the same dose that is given to people, and we actually in this study for the first time demonstrated through measurements in CSF that oxytocin inhaled through the nose gets into the brain. Okay, so we can see here in red that after inhalation of oxytocin compared to inhalation of saline, we get a major amplification of oxytocin signal in the CSF. So it's getting into the brain. What happens to behavior? We're looking at the exact same task I just told you about. 5 more minutes? Wow, okay. So, so this is the task in which monkeys can choose whether or not to deliver reward to another monkey or to themselves. And I just want to point you to this data here, which is broken out from the beginning of a session. This is for 2 monkeys over a number of different sessions, so there are thousands of trials in here. Gray is inhaling saline, and you can see that they basically prefer to give reward to the other monkey over no one, at about a 5-to-3 ratio. But after they've inhaled oxytocin, we see a major ramping up of prosocial choices. Okay, in this otherwise non-pair bonding, despotic macaque, we can actually make him nicer. He appreciates, he values what happens to the other animal more. And that may be tied to, just as we saw in, hinted at in that study of autistic individuals, that the amount of time that our donor monkey spent watching the other monkey drink the juice, okay, because he's allowed to freely look wherever he wants, also ramps up very quickly. And it's tempting to speculate that, in fact, this could be a very simple mechanism where oxytocin just turns up the volume, turns up the gain on the attentional priority, the value of looking at the other individual, and seeing that individual drink the juice becomes more reinforcing. And then that amplifies the behavior. There's much more to be done here. We want to understand how it actually works. So one thing we've been doing is recording in these various brain areas after the systemic application of oxytocin, but another thing is actually local injections of oxytocin. This is really too preliminary to even show you, but I just want to kind of give you a flavor of what we can do, which is that we can inject oxytocin, for example, into the anterior cingulate gyrus. This is that area where we saw those mirror neurons for reward. And when we do that, monkeys spend more time looking at the other monkey after you inject oxytocin compared to saline, and we see an increase in the firing rate of these mirror neurons when the monkeys are actually when the donors are looking at the other monkey. It's very preliminary, but it suggests the possibility that actually oxytocin is having an impact on behavior via this neural circuit. Now, I want to hopefully touch on the last bit of our talk, which is going beyond the sort of neurobiological mechanism and beginning to try to understand how these differences between individuals and species arise, what combination of genetic endowment and experience actually results in these differences in behavior. There's already data out there showing, for example, that various polymorphisms in different genes, like the promoter region for the serotonin transporter, is associated with kind of an exaggerated social fear response or anxiety in humans that's associated with increased amygdala activity. We see something very similar in the behavior of monkeys, where they show those individuals who carry the analogous short form of the serotonin transporter promoter allele show greater aversion to looking at other monkeys, especially the eyes of dominant males. But as you can imagine, doing genetic discovery in a laboratory where we have 16 macaques is not very powerful, given the thousands of genes that potentially we would like to query. So what we have done instead is to turn to a really wonderful natural laboratory in which there are over 1,000 rhesus macaques living on the island of Cayo Santiago off the east coast of Puerto Rico. This is a population that has been resident there since the 1930s. We know a lot about these animals because they've basically been under continuous observation. We know their pedigrees. We know life history data, when each animal left its natal group. We know the number of offspring they have. We know their dominance ranks, et cetera, et cetera, et cetera. It's a tremendous resource. And once a year, we have the opportunity to trap the monkeys and collect biological specimens. Actually, it's going on right now. So there are about 1,000 monkeys. We can get biological data on about 250 of them per year. And we have been concentrating on spinning out DNA in a small field laboratory that we have down there and then basically shipping it back to Duke. And one of the really important things about Cayo Santiago that's different from the primate centers here on the mainland is that it's basically minimal intervention. So the animals are provisioned with food, but otherwise there's not a lot of interference. So monkeys die on the island all the time. Monkeys kill each other. Monkeys fight. Some monkeys breed. Some monkeys don't. So there is the opportunity for evolution to be occurring here, right? So the behaviors that these animals engage in actually have some biological significance. And what we've been doing in terms of phenotyping these animals is going at this in a number of different ways, one of which involves painstaking focal animal follows, just as Robert talked about yesterday, when we have field assistants on the island following individual monkeys for 10 minutes at a time, and basically taking data on everything they do from about a 50-point ethogram of behaviors that are possible. And from that, we can construct all kinds of interesting measures, like the personality measures Robert talked about. I'll show you a little bit about some of the social network analyses we've been doing. We also get data in a variety of other ways. We can phenotype monkeys, for example, by challenging them with various stimuli, like going up to them and confronting them with a photo of a monkey from outside their group or from inside their group. And we can see whether that kind of challenge has any predictive value for the way that these monkeys actually behave in real life and whether there are any genetic correlates. So we just published a paper last week, actually, and this is the work of outstanding postdoc Lauren Brent, in which she has applied social network analyses, basically graph theory analysis, to look at 3 aspects of how individuals are connected to each other, and this is over 2 different years of sampling in one group amongst the 6 to 9 groups that are on the island. We can construct a grooming network, an aggression network, and just a proximity network, which is kind of who sits next to whom. So you can see that there are certain individuals who are in the mix, especially in this grooming network, who are very central. They're connected to lots of other individuals. There are some individuals who are completely peripheral. They don't groom with anybody. They're the complete outcasts of this group, okay, or they're the ones who are following. These aspects of how individuals are connected to each other, their affiliative behavior, their aggressive behavior actually has an impact on their fitness. So monkeys who are closer to the center of the group, who tend to have closer proximity to other individuals, have higher reproductive success than those who don't. And we also see an effect of aggression. That is, those individuals who are highly aggressive tend to have more offspring, but we also see a different strategy here, slightly significant, which individuals who have very low aggressive tendencies also have a slight uptick in the number of offspring they produce. The important point here is that we can actually, it appears that there may be evolution in action, okay? As I said before, we know how all these animals are related to each other from pedigree data derived from microsatellite information, and we can create these kind of maps that show this relatedness. The reason why the females line up along here where we're plotting them by their rank is because, as Robert said yesterday, like all old world monkeys, females inherit the rank of their mothers. Okay, so that's why we see that along the diagonal. And in fact, if we look at the pedigrees and we do an analysis of these, there are various measures one can derive from these social network plots, how many connections you have, how central you are, how strong your connections are. They basically tend to all be fairly highly heritable in this population. So if you are popular, if you have, if you're very central, if you groom a lot with a lot of other individuals, your offspring will tend to be that way as well. Your other kin will tend to be that way. And that's true even when we look at kinship through the male line, right? And males emigrate from their natal groups. We have, as a test case, just looked at 2 genes. We just pluck these, basically, out of the literature. We know that serotonin is important in terms of regulating mood. It's important in anxiety. It's been linked to autism, right? And so we have, just as an initial query, looked at variation in the serotonin transporter promoter region and also TPH2, tryptophan hydroxylase, which is the rate-limiting enzyme for producing serotonin. And basically, there is a minor allele for each of these, which is represented or found in only a few, in a small proportion of the population. And we tend to think of this allele as the one that is kind of unfavored, right? And it's for serotonin transporter promoter, it's associated with anxiety, depression, stress, being prone to alcoholism, et cetera, and similar pathologies for TPH2. And when we look at our network, we find that having only one of these kind of bad alleles doesn't really impact your connectedness in the grooming network, how kind of affiliative you are, how many grooming partners you have. But if you have both, that's a double whammy. Okay, and so it's these individuals, and they're indicated here in red, who have the minor allele of both of these serotonin-linked genes, who are really on the periphery of the grooming network. Okay, so these are the individuals who are not going to be doing so well, right? And they don't have a lot of connections. And we know from a lot of what we heard yesterday that these individuals are not going to have a lot of social support. They're going to be at risk for bad health, right, and bad consequences. Now, of course, 2 genes aren't the story. They never are. There are thousands of genes, and so we are beginning to tackle that question in a couple of different ways. One is that we have created a SNP array, and there's about 301 SNPs in this, in a number of different pathways that we think might be important for behavioral function. These are different groups, and each of these are individuals, and the red are having the minor allele for each of these different SNPs. And then we're also beginning to do whole exome capture to do more or less full sequencing for these monkeys. And then we have also begun to try to parse how these phenotypes, these sort of abstract networks, actually relate to the behaviors themselves, the traits. Because being central in the network is not something that really can be directly inherited, that is somehow a product of the way that you interact with other individuals. So it's some combination, we might imagine, of personality, temperament, and social skill. And so we've begun to do a lot of the same kinds of analyses that Robert talked about yesterday to begin to look at those aspects of the phenotype. And I want to just leave you with the parting realization, admission, that of course it's not all about genes, right? And that experience has an important role to play, and the context that you're in has an important role to play. This is some data from a very beautiful paper from Jerome Salin and Matthew Rushworth that came out last year, in which they took rhesus monkeys and they put them into different size groups from 1 to 7. And they let them live like that for something like 9 months or a year. And then they did structural MRIs on these animals, and they found that areas that had been implicated, like in the superior temporal sulcus, areas that had been implicated in social function, social identification, recognition, etc., changed. And they varied as a function of the size of the group that you lived in. So those individuals who lived in bigger groups had thicker gray matter in these social processing areas. And they also showed stronger functional connectivity between these areas as well. So that suggests that, in fact, it's not just the biology that you come to the world with, but it's also your experience and the context that you're in. So if, for some reason, a hurricane wiped out half your group, that would also change your underlying neurobiology, and that would presumably have a change, that would cause a change in your behavior as well. So I know I'm over time, and I'll just leave this summary slide up and go ahead and take questions.